Hello, bachcho. Let us look into the question. The question says that in Meselson and Stahl's experiment that was carried out in 1958, DNA extracted from a culture one generation after the transfer from N14 to N15 to N14 medium had a hybrid or intermediate density Y. So these are the options given to us. Let us look into it. The key concept is that Meselson's and Stahl's experiment. जो conclusion हमें देखने को मिला that replication is carried out and it is in the form of semi-conservative replication. Now what was the experiment? Let us look into it. First of all, there was introduced an isotope of nitrogen 15N, alright, and this was made to be inculcated into the genome of E. coli. For numerous generations, this was introduced into the genome of E. coli so that ये बिल्कुल उसमें present हो in the form of N15 itself. Now this N15, it was introduced into the genome of E. coli having N14, and at this stage what we saw कि जब हम centrifugation carry out करते हैं तो we see a band that is obtained. It has a intermediate density and it was of both N4 and N15, and the generation time of E. coli for the replication was 20 minutes. After that, in the second generation, what we saw that two bands of light DNA were obtained. Along with that, two bands of intermediate DNA was obtained. That means two bands of intermediate DNA and two bands of light DNA, यानी कि normal form. Since heavy DNA had N15. So ये था Meselson and Stahl experiment जिसमें प्रूव किया गया कि replication is semi-conservative as we have seen in this experiment. So let us move to the options. हमारे option है because the generation time of E. coli was about 20 minutes, because it would take 20 minutes for RNA replication, because it would take 20 minutes for replication of DNA to RNA, because it would take 20 minutes for translation of RNA to protein. As we know, during the DNA replication what happens, DNA से ही DNA बनता है, यानी कि parent DNA के strand से offspring DNA strand बनता है, right? A new DNA strand is made. So it is only talking about DNA itself, and there won't be any RNA, there won't be protein, there won't be transcription, because transcription central dogma में replication के बाद आने वाला process है. So the statement that is correct for the formation of this hybrid after one generation, it is because the generation time of E. coli was about 20 minutes. This is a right statement. Let us conclude it. Option A is the correct answer. Hope you all understood. All the best.